Thank you. Remember to follow us on LinkedIn. We only got like 30 new followers. We are looking for more, if you please. Okay, thank you. Let's start with the last session we put together for you today that hopefully will bring up a lot of discussion. So we're going to talk about controlling FMD using novel vaccines. We have two speakers to kick off the discussion afterwards. So we start with Danny Goovaerts, please.
Okay. Thank you. I would like to thank the organization for allowing me to give you this presentation and to share my thoughts with you. So my presentation is going to be about, well, we all have heard about conventional FMD vaccines and controlling the disease. But I'm going to focus on, why would we, if these vaccines are available, why would we still need some new technologies? So just the content, I'm going to briefly say something about the current vaccines in general. Then I'm going to focus on what could now be the limitations of these vaccines and where could we possibly improve and what are the bottlenecks also in manufacturing. And then what should then the new technologies bring you and some conclusions. So just briefly, the history of the foot-and-mouth disease vaccine development. There's a couple of milestones. An important one, I think, is in around 1960, where the virus started to be grown in BHK suspension cells. Why is that important? Because it first allowed, let's say, large-scale manufacturing. If you have suspension cells, you can grow it in a biofermenter, and that made a big difference instead of growing it in flasks up to them. Another very important milestone is in 76 with the introduction of BEI as the inactivant, because up to then there's been many problems. People were using formaldehyde, and that very often resulted in incomplete inactivation. But if we look to the process as it was then, 40, 50 years ago, in essence, apart from some NSP purification which has been added, there has been very little or almost no change in the process as such. So if we look where FMD manufacturing is done all over the world, then except for the US and for Australia, where you're not allowed to work in Australia, not at all, but where there is no permit, let's say, or no possibility to work with live vaccine manufacturing on the mainland. So basically every region has smaller and larger manufacturing facilities in the world. If you look to the product profile of the current FMD vaccines, it's relatively straightforward for an inactivated vaccine. One important aspect is that to manufacture the vaccine, we use wild-type virus with all the biosecurity consequences. But it's oil adjuvanted, although you can also use gel vaccines, but most of the vaccine for matters of duration of immunity and impotency are oil adjuvanted, double oil emulsions, single oil emulsions, duration of immunity of 6 months. So it's a very straightforward inactivated vaccine. And we've seen also presentations during these past days. It's a great tool in controlling the disease. Without any doubt, they have played an important role to control the disease, to eradicate the disease, like in Europe, South America, yeah, you can talk about is it already eradication or not. But anyway, the disease is very much controlled over there. And we've also have seen that on some occasions, clinical outbreaks of the disease can be very quickly controlled once we start vaccinations. And I have a nice, well, probably many of you have already seen this slide, but it's a nice demonstration of the efficacy on clinical disease control of FMD vaccine. In 2010 in South Korea, there was an outbreak of FMD. Initially, the government tried to control the disease by stamping out an eradication. But after a couple of weeks, they realized this was not going to work. So they started vaccination. And then you can see the blue arrow where they started vaccination in the cattle. You can nicely see that after 2-3 weeks, the peak on incident of the disease was already declining. And then a couple of weeks later, they started vaccination in the pigs. Same story, after 2-3 weeks, the number of clinical cases very quickly dropped, and the disease was actually, well, under control. Not completely, but definitely the clinical cases were. So it clearly demonstrates the efficacy and the use of these FMD vaccines in clinical outbreaks. So the question is, if we have these vaccines, why should we still need new technologies? If we are able to control the disease, we can manufacture them, we already know for 40, 50 years on how to do it, why should we still need better vaccines? And the thing is that despite the fact we know perfectly on how to make the vaccine, people don't really realize, people in manufacturing know, but most people won't realize that to manufacture good FMD vaccines, it's a big challenge. And the challenge is high, and it's frequently underestimated. I've been working in my career in the development of many, many different vaccines, and I consider FMD vaccines still one of the most challenging vaccines to make. And that is, unless you are okay with substandard quality. Then it is easy, and that is also often the case. That also happens. The people who were here 2 days ago, heard the presentation, a very nice presentation of Susan from Uganda, where she looked at post-vaccination monitoring of 2 different vaccines. Now, one of these vaccines just showed a flat curve, no response after the boost, no response after the primary vaccination, no response after the boost. And that is a challenge. So if you look to the limitations, I split them up in limitations of the current FMD vaccines, which you could say, which are actually directly virus-related, maybe vaccine-related, but also manufacturing aspects. And I'll focus most on the manufacturing aspects in the rest of my talk. So the virus-related aspects, of course, we have a number of serotypes, a number of strains, and there is a lot of variation even in the manufacturing aspects between the strains. We all know capsid stability and antigen stability is a challenge with FMD. And then there's also some political and security aspects, which are the direct effect of using wild-type strains in a certain area for vaccine manufacturing. Immunogenicity of the current vaccines could be better. The duration of immunity is 6 months, which is, and although you get very nice clinical protection, you actually don't have a real effect on the carrier status. So also there might be some challenges or some possible improvements. But I'll mainly talk about these aspects. So we use the fact that we use virulent virus to make the product has a number of serious consequences. But one of the main consequences is that we need high containment facilities to manufacture the vaccine, so-called BSL-3. So capital expenses, if you want to put a new manufacturing facility in place, capital expenses are huge. As a ballpark, it will of course depend on whether your equipment, the region that you build, also whether the equipment is maybe Chinese, Indian, European, US derived, that might make a cost. But generally you could say, if you want to make a facility, the price roughly of your capital expense is going to be $1 per dose that you want to manufacture. So if your annual capacity has to be 100 million, you're going to build a facility more or less in the order of $100 million. The capital expense is one, but there's also a very substantial operational cost during the manufacturing of FMD. And I'll come back to that. It has a couple of reasons. So in FMD, you have to operate at very large scale. And you have to do it very, very efficiently. One of the reasons is that, well, I would say that building a manufacturing plant for something less than 50 million doses of vaccine annually or antigen is actually not really competitive in the field. Because a very important aspect is you don't really have the economy of scale and you will have a very big problem, let's say, of making it meeting an acceptable cost of goods. The limitation of building these high facilities, there's also other consequences. Most of these facilities are purely, entirely dedicated to FMD manufacturing. It's almost impossible because you're going to need a lot of validation because you work with wild-type virus in these facilities. If, for instance, you want to use them to manufacture another vaccine, it's almost impossible. You need a big validation step in between. Another consequence is that if you would ever stop vaccination, like what is happening now in Brazil, it's very difficult to find another use for these facilities. Because of the size of the fermenters, there's very few processes that you can fit in it. And that also plays a role in the expense. And to make them profitable, you need to operate them basically on 7 days a week, the whole time, every day making FMD batches. Otherwise, it's going to be too costly, or capital expense is going to be too costly. So that operating them on a continuous basis is also limiting your flexibility to respond to surge and demand and capacity demand in case of outbreaks or sudden request for vaccine doses. So just a picture to give people an idea on this is a media preparation area of such a facility. You can immediately see a little bit the size of the fermenters. Oh, it's not fermenters, of the vessels, let's say, the mixing vessels. So economy of scale, just to give you a bit of an idea where that comes from. For instance, if you compare a 5,000-liter fermenter and a 1,000-liter fermenter, basically the price is not very different. So of course, 5,000 is more expensive than a 1,000-liter fermenter, but the price is not so different. Why is that? Because on both fermenters, you're going to need the same probes, the same ports, the same motor. You're going to need to have a software system, so the controller. And the only difference between a 5,000 and a 1,000-liter fermenter is basically the amount of stainless steel. Also, as a footprint of a 5,000-liter is not 5 times the footprint of a 1,000-liter. So if you build a bigger facility in capacity-wise, it's not gonna, your total facility and your capital expense is not gonna multiply in the same order. Another aspect, let's say, which is gonna influence your cost of goods, if you have a capacity, if your capacity of manufacturing goes up, your purchase price is also gonna increase. So you will have more negotiation power with your suppliers that should supply the raw materials or the equipment or everything, if you're going to be able to say, okay, I'm going to produce 1 million doses, I'm going to produce 100 million doses. And that will also have an effect in your cost of goods. Quality control, if you formulate a vaccine. If you can formulate a vaccine like at 1,000 doses or 5,000 doses per batch, it's the same quality control. So the cost of your quality control, if you can divide it by more and more doses, is also going to go down. So that is why the margins, if you don't produce at high scale, are going to become very narrow. And FMD vaccines is a very, very competitive market from a cost of good perspective. And if you don't have everything perfectly under control, you're going to be in trouble with the cost. So I already said that. Well, there's other aspects of using virulent virus, and that is more of a what you could call political. Like if you're in a certain country, if you have a manufacturing facility in a certain country, let's take the example of Brazil or the example of India. These countries are only going to allow you to use local strains because of the biosecurity risk. And that, of course, also can limit your possibilities of using the vaccine also for export. Also, that can be a problem. Okay, and then we have, of course, one last but not least aspect of using virulent virus, and that is the risk of breaching the biocontainment. So if you ask yourself what is actually common in these different places, they are all nicely touristic places. Well, some of them maybe are. But these are all places which had multiple documented outbreaks in the past, either because of escape from the facilities or because of failures with vaccine inactivation. You can see that a lot of them are very well known is the last outbreak in Pirbright of 2007. But at the time, let's say, that Europe, we hear that somebody in the morning also making a remark about it, when every individual country was making their own vaccine, that we had multiple outbreaks in Europe, and this is also a demonstration of that, I think. There you also see that between 69 and 96, 25% of all primary outbreaks could be linked to vaccines. Either the manufacturing, the escape from the facility, or improper or not full inactivation control. So it's a serious risk as well. Then another aspect, which is very, very important and that people won't really realize, is that manufacturing foot-and-mouth disease is a very it's a difficult vaccine also because the yields are actually low. If you see what you get, and these are, of course, it's depending on serotype, on strain, it will differ, but these are like average ballpark titers. Then you could say if you get 3 microgram upstream, will be relatively okay. You will, of course, you might lose some depending on how well you manage your downstream processing, but let's say you could use something up to 30%, that's not really exceptional. So you would end up with, in your concentrate, at the end, 2 microgram per milliliter of, compared that 2 microgram of 146S antigen compared, derived from 1 milliliter of virus culture fluid. To make a good quality vaccine, again, depending on the serotype, it's going to be a bit more for O than a bit less for A, but an average 7 microgram 146S per dose is certainly not out of the range. So that means that if you would want to make a trivalent vaccine, let's say an Asia 1 for India as an example, you're going to end up, if you want to make a good quality vaccine, you're going to end up with something like around 21 microgram in one dose of vaccine. And that actually, with 2 microgram from 1 milliliter of virus fluid, that actually comes from, you're going to need 10 milliliter of BHK fluid to produce that. That is enormous. That is enormous compared to many other vaccines. And that is a major aspect, a major reason why everything in your manufacturing facility needs to absolutely optimal working, because otherwise you're going to have immediate problems with the profitability of your operation. So to illustrate it a little bit better, I made a table, let's say, where I compared our FMD vaccine, our inactivated trivalent FMD vaccine, with live PPR. If you compare, like, well, as we said, like, where's the pointer on this? Okay. So for foot-and-mouth disease, you're going to need 10 milliliter to make 1 trivalent dose. PPR, we can grow it in manufacturing up to something like 10 to the 7. Let's say we lose a log somewhere in the downstream. The minimum protective dose is 2.5 TCID50. Of course, to compensate for stability and for freeze drying, you're going to put in a bit more because you don't want to target the 2.5. So we target 4 TCID50s. That means that for PPR, out of 1 milliliter of virus fluid, we're going to get something like 100 doses easily. So, and for FMD, we're going to need 0.1 dose out of 1 milliliter. If we want to make 1 million doses of foot-and-mouth disease vaccine, we're going to need 10,000 liters of virus fluid. For PPR, that will be 10 liters. And then looking to the serum, which is also very often a big problem, to get decent supplies and sufficient supplies of bovine serum, depending, of course, on how much serum you use in the medium. But if you use 5 to 10% serum, for this 1 million doses, you're going to need 500 to 1,000 liters of serum in your FMD, and half a liter or a liter for the PPR. So that's the FMD, of course, you're going to have to produce it in roller bottles, in fermenters, while the PPR, you can still get away in a roller bottle. Now, if we take India as the example with these numbers, India has plans, let's say, or is in phase of doing a big control program with 450 million animals who have to be vaccinated twice a year. We're going to end up with like 900 million animals to vaccinate. The amount of cell culture fluid you're going to need for that equals around 9 million liters. Same amount virus fluid, 9 million liters. The serum you're going to need for that is, depending on use 5 or 10%, it's going to be 500,000 to almost 1 million liters of serum. And if you run your operation, and I told you you have to run the operation. Okay, I'll go quickly. If you have to run the operation on a daily basis, 300 days a year, because otherwise you won't be able to recover your investment. You're going to have to produce on a daily basis about 30,000 liters of virus fluid. So that's the size of the operation, which makes it very difficult. Now, some of the virus, just quickly, we already heard that. We got presentations in the EU FMD on virus particles falling apart in 12S. So the virus particle is very unstable. I don't have to explain that, I think, and there is many different events in your manufacturing, the pH, the temperature, the time, the shear forces that you need to have under control, otherwise you're going to lose antigenicity and antigen in your product. So minor details in the manufacturing can make a big impact on your antigen stability and on your vaccine stability. Okay, so shelf life, I'll skip that. So conclusion, although FMD is a very old and a well-established process, If you want to make good quality vaccine, then it is, in my opinion, still one of the most challenging vaccines to make. Because we should also not understand that is in case the manufacturer still wants to make some money in making the vaccines. So, last slide. If we consider all of this, what should be the focus? If we think about new technologies, what kind of problems should be solved by these new technologies, and what should we think on? There's possibilities if we want to make, if we find an alternative to virulent virus, and BSL-3, you could think, oh, we could make maybe VLPs, attenuated live viruses, vector approaches to increase the output. You could think on modified master seeds maybe. Also maybe VLPs could also give you a higher output. Stability, we can, by using other technologies, if we can increase the stability, like we've also heard some presentations these days, if we could increase the stability in some ways, that could also be very helpful. Immunogenicity could be better. Cross-protection and DOI. So, thank you very much.
Thank you, Danny, for the summary of where we are with conventional vaccines. Thank you. So I'm Toby Tuthill from Pirbright. I'm going to be co-moderating this session. So we'll move on to the next speaker, Giselle Medina, who's going to give us an overview of progress towards these novel vaccine approaches.
Good afternoon, everyone, and thank you for staying to the last presentation today. And thank you to the GFRA committee for inviting me to give this presentation. So today, my job is to summarize some of the current and advancements in the novel vaccines for FMD. So these novel vaccines are composed of virus-like particles, either in the purified form or those that are delivered by recombinant viral vectors, peptides, DNA, or mRNA, also known as genetic vaccines, and modified live attenuated vaccines. All of these approaches are dedicated to address the challenges of the current chemically inactivated vaccine in an effort to reach the ideal vaccine for FMD. And looking at these parameters, this vaccine should be safe, it should have temperature stability, it should also give long-term immunity to these animals, it should be cheap, it should also address the problem with persistent in ruminants and maternal immunity, and it should have a DIVA and, of course, confer protection against different serotypes. Importantly, not a single approach for these novel vaccine platforms will meet all the criteria for an ideal FMD vaccine. I'm not going to go over the advantages and disadvantages. I think at this point everybody knows what are some of the key problems with the current inactivated vaccine, but I just wanted to highlight some of the key progress that has been made in this vaccine platform, starting with the improvements in capsid stabilization. There's been efforts in identifying antibodies that can recognize the stable capsid, and these antibodies can recognize against all the different serotypes. There's also been efforts in improving multivalency and cross-protection, and also the development of chimeric VLPs and mark-inactivated vaccines. One example of this is the leaderless vaccine. Here, the leader protein coding sequence was removed and also incorporated a few restriction sites that allows for the easy swap of capsid sequences, and in addition, the incorporation of the DIVA markers. There's been an emerging technology, these mosaic vaccines that are developed by evolving sequences using a computed algorithm, and this has a lot of promising as a vaccine platform. Next, I'll move on to purified VLPs. We have heard several talks in this forum on the great results on the VLP technology. It works great because these are self-assembled. They can mimic the size, the shape, and the symmetry of FMDV. They are easy to produce, and they're able to induce humoral and cell-mediated responses. Some of these disadvantages are the potential instability, the need for adjuvants, and the require and boost vaccination strategy. And of course, the production can be complex. On the right-hand side, I have made a table where I have categorized the production of VLPs based on the expression systems that have been utilized. So for bacteria, some of the advancements have been the incorporation of amino acids that will enhance the immunogenicity and also the yield. There has also been studies where this VLP can be combined with other vaccines, or multiple serotypes can be incorporated. And also, because production can be problematic, there has been changes made to increase the water solubility and also the inhibition of protein aggregation. In yeast, 2 platforms have been used, the Hansenula polymorpha and Saccharomyces cerevisiae. These VLPs have been tested, and it looks like they are doing great. However, this work has been done in mice. In mammalian and baculovirus expression systems, there has been the greatest advancements, where there has been multiple modifications to improve the thermal stability and also the acid resistance. And we cannot forget plants. Plants are also being considered as a potential platform for the development of these VLPs. Next, we have the recombinant viral vectors, and these are not really the purified VLPs. These are vectors that encode the P1 and some other FMD antigens that all contribute to the formation of VLPs. These VLPs are made in the host. One example of this recombinant viral vector technology is the use of the human adenovirus vector vaccine that provided great results in cattle and swine, and that culminated in the licensure of a candidate vaccine in the US for emergency use in case of an outbreak of FMD serotype A. Most recent, there has been progress done by developing this adenovirus with the FMDV3D antigen, and this has been tested in mice with great T-cell responses. And also the combination of the adenovirus FMD with the adenovirus encoding different type of interference. This in an effort to shorten the window of vulnerability, since the vaccine requires at least 7 days for the adaptive immunity to kick in. So advantages of this vaccine is that they're DIVA, they're composed of FMDB MC capsids. They can induce antibody and cell-mediated immunity. And importantly, it does not require a BSL-3 lab for production. There's, of course, disadvantages. It's expensive. It does require a large amount of these viruses. It does protect at 7 days post-vaccination, and the duration of immunity is not that long, and also it does not induce cross-protection. There's also other recombinant viral vectors that have been explored, including vaccinia virus, foulpox virus, attenuated pseudorabies virus, and Semliki forest virus. All of these studies have used different vaccination schedules and all tested in different animal models, but also in the natural host for FMD. In particular, this single-cycle Semliki forest virus does look promising. However, there's this theoretical safety risk for recombination with other alpha viruses that may be circulating in nature. I'll move on now to peptide vaccines. These vaccines are dominated by the incorporation of these immunodominant B or T cell epitopes, which are crucial for protective immunity. And usually for these peptide vaccines, these epitopes are arranged in tandem, so the more epitopes, the better, with ultimately facilitating this MHC1 or MHC2 presentation. Advantages of this platform is that it's very specific, it's safe, they're compatible with the use of adjuvants, and it can be cost-effectiveness. Some disadvantages is that it could have a low immunogenicity, so that's why it may require an adjuvant, and the epitope can vary. Some of the most recent advancements is the use of dendrimers and multiple antigen peptide vaccines. These have been shown to elicit a strong immune response in pigs, and the protection can last up to 5 months. In addition to that, there has been combinations between peptide vaccines and other immunomodulators like poly-IC and nanoliposomes. Most recently, this Addamer technology has been utilized. The Addamer requires certain proteins of the adenovirus type 3. They self-assemble as a nanoparticle and then serve as a scaffold for displaying multiple FMDB epitopes. And most recently, these recombinant T4 phages have been used to present the BP1 B cell epitope. However, for these last two, the studies have been done only in mice. Moving on to DNA or RNA, also known as genetic vaccines, and also known as the third generation vaccines. These are known for being safe, easily to produce. Mainly for DNA, they may have a low manufacturing cost and long shelf life. Disadvantages: low immunogenicity, may need large amounts, and multiple vaccination schedules. And of course, for DNA, there's a potential risk for integration. This vaccine platform requires or is supported by transcription and translation processes, all in favor or facilitating the link to the MHC1 and MHC2 pathways. Some of the progress that have been done in this vaccine platform is the increase of efficacy DNA vaccines through electroporation and also the combination with cytokines, genetic engineered vectors, liposomes, and other nanoparticles. For example, the design optimization in plasmids that are expressing FMDV antigens, there were specific changes in the 3C coding sequence so that expression of that protein is not toxic, and that resulted in great immune responses when tested in animal models. This is another example where the plasmid DNA vaccine was combined with a cytokine to enhance immune responses. And there's a few more examples of the creation of FMDV mini genomes that can potentially be used as plasmid DNA vaccines for FMD, although they have not been tested in vivo. And finally, the combination of use of VLPs plus plasmid DNA encoding some of the FMDV antigens. And of course, we cannot forget about the live attenuated vaccine, since it has had great success in eradicating diseases for other viral diseases like rinderpest and smallpox. In the context of FMDV, there has been some studies looking at this possibility. In particular, the study where 2 amino acid changes were done in the L-Pro generating a submutant virus that was attenuated in vitro and in vivo. And when we evaluated this vaccine candidate, we saw complete protection in swine. However, because there were only 2 amino acid changes, there's a high risk for reversion to virulence. For this reason, we have utilized the colon de-optimization technology. This has been shown previously to affect the efficiency of gene expression. Basically, we can replace the natural codons with non-preferred codons or shuffling of codon or codon pairs within a sequence, and this can dramatically affect its expression. We have several reports of this technology where we have colon de-optimized almost the entire genome of FMDV and still acquire viable virus. One example in particular is the P2P3 colon de-optimization, where we have incorporated 687 nucleotide substitutions. And again, this did not result in amino acid changes. And I'm just going to briefly share some of the data. We inoculated cows with different doses, and we were looking for safety and efficacy profile of this vaccine. None of the animals that were inoculated with our colon de-optimized vaccine resulted in disease. We also looked at neutralizing antibodies, and when compared to the control, there in yellow, you can see that all of the animals vaccinated had a very robust immune response. We look for ELISA, because this vaccine has a DIVA, and indeed, this vaccine is compliant for DIVA markers. And we also evaluated T-cell response after challenge, and we can see that there's a powerful, strong T-cell mediated response at 5 days post-challenge. So advantages of the live attenuated vaccine is that they're cost-effective, they can induce humoral and cellular immunity, they can also enhance broad cross-protection, at least with this colon-optimized vaccine. And it will reduce the need for adjuvants. Disadvantages, of course, there's always the risk of recombination. We don't know if this vaccine will address persistence in ruminants. It will require cold chain maintenance, and it will have a difficult time with the regulatory agencies as well. So conclusions, novel vaccine platforms offer to address the limitations on the current activating vaccine. Oh, this is not my,
Go back.
I had more, yeah. Anyways, I just wanted to ensure that some of these novel vaccine technologies have been tested in mice, and I think there is a critical need for a very thorough evaluation in the natural animal hosts so that these vaccines can go to the next level. And of course, these vaccines have also need to be tested perhaps in field conditions and where the animals are also stressed, and those animals may be dealing with co-infections as well, and testing whether these vaccines are efficacious in those conditions. And with that, thank you so much.
Thank you, Giselle. So now if I invite Danny and the other panelists to come up to the table, that would be great. So we're joined by Jacqueline Horsington and Elizabeth Reeder. And I think David McKay has also kindly offered to give us some regulatory expertise. So if you want to come up, David, you're welcome.
Is there another chair?
Please.
Yes, here's a chair.
Oh, yeah, yeah. This one. So this is the discussion forum where we're going to get global agreement on what kind of approaches we need for the future for vaccination to control FMD. We're going to get complete agreement.
No, that's fine. There's no more space for you.
So just to preface the discussion, the key questions that we might come back to are conventional vaccines good enough, or should we be considering some of these other technologies? Regulatory issues, what are the hurdles to introducing new technologies? And the third of my questions is, what do we need to do to drive industry to take a role in this? What's the drivers for industry to make different vaccines? Those are my three questions. I'm sure there'll be others, but...
Yeah. No, but we can start with the industry. Maybe we can give the microphone to you.
Oh, you want the three questions?
No, you can address the one you want, the last one.
Where do we start? I think it was clearly stated. Current vaccines have limitations. Clearly, the industry is interested in new candidates, and we receive a lot of very juicy, promising offers of the FMD vaccine of the future that will solve all our problems. But it's more complicated than that. It's a difficult virus, and the first criteria that a candidate has to pass it needs to be as good as the existing vaccines. And many of these candidates have limitations that make them unfit for the market. Let me give you an example. We looked at several technologies. We played with a lot of them. There was one that looked really promising. We had very good efficacy in target species, in cattle. And the cost of goods looked compatible with large-scale production. The process could be industrialized at large scale. So good. But then we tested it in pigs, and it didn't work. And then we tried 2 strains together. It didn't work. There was interference. That killed it. An FMD vaccine needs to be combo, and it needs to work in both species. So that killed that very nice candidate. And so something else that is not in the mind of everyone is that, as my colleague mentioned it clearly yesterday, once you have demonstrated that proof of concept, that this is a nice candidate, there's a lot of work that remains ahead of us. 5 years of development, millions of dollars to demonstrate, to develop analytical tools, to scale up your production into these thousands of liters, to demonstrate stability, which takes a long time, to do all the clinical testing of each of the strains in the target species, demonstrating duration of immunity, which takes an awfully long time, producing 3 consecutive large-scale production batches, satisfactory release, and then registering that product in multiple countries. And Europe is not the benchmark, is not the model. In Europe, you submit 1 dossier, it's evaluated centrally, they give you a yes or a no. In Southeast Asia, each country has a different regulatory body. They each have a different set of requirements. So sometimes you need to do a study just to satisfy the needs of Vietnam. And some countries have not even incorporated the multi-strain approach. So if you change you have a 5-strain vaccine, you change 1 strain, you need to repeat everything for all the strains. So I'm sorry, but this is hurdles we have to face. This is what we have to do after we are presented with these nice prospects. And that's how we screen the candidates and decide to go or not.
Thanks, Pascal. I think this is really useful for people to hear what the real challenges are, what the real hurdles are.
No, I just want to add to the comment, the recent comment that, or come back to you with the question: is the current vaccine quality the bottleneck to improve the control of the FMD worldwide, or the current vaccines are able to control? Because it's already demonstrated in every countries or regions that the current vaccine quality are enough to control the disease. So where we should invest or put our efforts in the next year?
Yeah, I think one of the problems is also, and I didn't even mention it that much in my presentation, I think one of the problems is also like, take the developing world. If you look to, let's say, Africa, there's a huge amount of cattle in Africa. FMD is endemic almost all over the continent, I think. And the total vaccination ratio in Africa is around 5%. If you have well, we heard Nigeria several times during the conference. If you have to vaccinate 150 million cattle twice a year, and then the prices of the vaccine in Africa are relatively high, so we talk about $2 a dose, that would cost Nigeria around $500 million annually. If they have to run a control program for that time, I honestly don't think they will get the money. So in other words, if we don't do something drastically, and I couldn't agree more with the manufacturers that licensing vaccines takes time, and it is complicated. But if I would have a look to, let's say, from a technical and from a regulatory perspective too, just as an example, the live codon D optimized, it sounds, a lot of people will say, whoa, but a live codon D optimized vaccine, of course you will have to demonstrate it is stable. Of course you will have to demonstrate it doesn't revert to virulence. But I think with such an approach, in my opinion, that should be possible. That's what I would expect, at least. If with such an approach you could lower you could lower the need, let's say, and instead of we can grow the FMD up to 10.7. If you could get away with 10.5, and now that could have a major impact on the price of the vaccine and still have a profitable margin for the manufacturers. That's my personal opinion. So, yeah, I know FMD vaccines are good, but the problem is, well, in India, we have been in the past, we have producing a good trivalent, a good quality trivalent vaccine. 10 years ago, the price was around 11 rupees. That's 12, 13, 14 dollar cents. The result was that we were just making a loss on the manufacturing. So in other words, I think we have to, there is an urgent need, let's say, to come to something which, which could increase the output and making the vaccines more at a profitable price. Yeah.
Okay, so Danny's taken us straight into the most extreme alternative novel vaccine approach that we can discuss. I mean, just if we just link into that, the biggest thing that people talk about with a live attenuated vaccine is that the regulatory issues around the hurdles are too great. It's never going to be possible. And so this is where I might ask David to have some comments.
Yeah, and I think that's often quoted, and I think that the challenge is that often when people are developing it, they think that there'll be a kind of tick box approach to safety, and therefore, if I can show this at this level, this at this level, that at that level, then it'll be deemed safe. But unfortunately, it doesn't really work that way, because it's always done on a benefit-risk assessment. So that may be qualitative, it may be quantitative. So yes, you have to demonstrate safety to the animal, the user, the environment, and the consumer. So you have to look at each of those ones separately, and so you can demonstrate safety to the animal. The challenge with the live attenuated will be around the environmental risk assessment and the risk of transmission to non-target species and to the environment. And there, really, you just have to go through with effectively, certainly to start with, a qualitative risk assessment and generating the data through the studies to show and to quantify what the actual risk is, because that's what you're going to be asked. But I think what researchers, as I say, are often asking for is, well, what level do I need to show? And it's not quantitative. You would have to do a qualitative risk assessment and show that there is, and certainly with foot and mouth disease, the threshold is going to be very high. So as I say, it's not a checkbox, it's a benefit risk assessment.
Yeah, go.
I just have an opinion. So I think one of the advantages that the new technologies are bringing compared to the conventional is that there is a possibility to reduce the cost and make it more accessible to the endemic world because some of these technologies have the possibility to be produced in a BSL-2, so you don't need a high containment. And an example is the vaccine platform that we have been working in the last 10 years of a highly attenuated strain that is DIVA compatible. So you could produce a vaccine that may potentially have low cost because you don't have to undergo these extensive or expensive manufacturing processes aiming at eliminating the non-structured proteins and making DIVA compatible. So you could actually produce a crude PrEP inactivated, and you could have a product that potentially could be done in a BSL-2 and with less process involvement or expenses associated with that. So that offer an opportunity to have something more available for the endemic world. So that's I wanted to, and also regarding the attenuation, just one comment is that in our experience working for almost 20 years on recombinant vaccine and attempting to look at the safety and the development of attenuated strains of foot and mouth disease, the problem is that you go too far and attenuated to an extent that you don't even have replication on the host, like is the case of the leaderless viruses, or you have something with the potential to reversion and could go back to a level of recombination or be potentially recombining with other viruses, not different, but other FMDV, and recover the virulence by reversion or recombination.
So does anyone else want to continue with the theme of live attenuated while we're on this subject?
No, just a comment, but the fact is that in human vaccines, most of them are live attenuated. And sometimes it breaks my mind why it's so complicated in the veterinary field. We are still using a lot of inactivated vaccines, and the live attenuated in the human world have been used forever. So, of course, it's not my area. I'm not a vaccine producer, but I would like to know, even regulatory, why? Where's the difference?
But there's an enormous number of live attenuated vaccines used in veterinary health. No?
Yeah. Not the same amount, I think. There's a lot of
I
inactivated.
think a large number of all the poultry vaccines are live
Poultry,
attenuated.
yeah.
Huh?
So it
We
wouldn't
have
be that
pox,
difficult
lumpy
to
skin, PPR, Rift Valley fever vaccines, and there is an enormous number, an enormous number. And one of the big advantages, I know, of course, safety is then something to look at, but one of the big advantages of live attenuated is, of course, the dose and the price. So you can get much more, well, because of the virus multiplies in the animal. You need much lower doses of antigen to produce such a vaccine. I would also like to say something on recombination. Very often you hear like, yeah, but if you have recombination, then it is dead. I disagree to that. And the reason is that I'm part also in the Netherlands of the Committee of Genetic Modification, and that's the committee who advises the government on introduction of GMOs in the field. And the field can be animal experiments, can be in a clinic, can be release of a licensed product. And how we look at recombination is we don't see recombination as an absolute showstopper. No, we look to what is going to be the result of, what is the risk of a recombination? Are you going to get, because if you get a recombination, you have the wild type virus, you have the wild type virus already there in the field. The only question is, with that recombination, is the end product going to be more virulent or more problematic than the strain who is already there? And if that is not the case, then we don't really consider it as a problem. So that makes a difference.
Yeah, go.
So just a good discussion, but Danny, in your presentation, I suppose one point you're missing is, and you didn't mention, I don't think, anything about Nagoya. So it'd just be a comment, perhaps thinking about access to new strains and the importance that's becoming increasingly important for production and as an obstacle if we're putting a comprehensive list together. But I suppose just extending this discussion about novel technologies, and it's sort of more a challenge. I think there's often an easy way that we compare with what's been successful in South America, we compare what was previously successful in Europe. And there, of course, we say, well, the existing vaccines are okay, that's what worked previously, and all we need to do is to roll them out in scale. But of course, the challenges of controlling FMD in the current endemic settings are much more significant. And I suppose it's a question, perhaps, to those of you that have more experience of livestock in endemic settings, is the extent to which the current vaccines require that you have continuous vaccination over long periods of time to maintain protection and protective immunity. And the other issue, of course, is you have a lot more diversity in antigenic space than exists in Europe and exists previously existed in South America. And again, that poses a lot of challenges for the existing vaccine technologies. And I suppose that should be perhaps focusing our attention more than just immediately thinking the problem is the quality of the vaccines is poor, we just need to put them out at scale. And of course, a link to that is the cost and shelf life and all these other things are critical. But I'm just wondering, are the two factors, duration of immunity and ability of the product to generate a broad spectrum of immune responses that are protective? Are those the two criteria for the products that we should be focusing on?
Thanks, Toby, Medina, and Danny. Yeah, Danny, I must especially thank you for bringing to life real-time situation that is so much applicable to the African perspective. Thank you. What lessons can we learn from COVID-19 vaccines? It took just a very short period for vaccines to be rolled out globally, even though the principle of vaccine equity was not respected. I am a member of the African Vaccine Manufacturers Initiative, and currently there are initiatives or there are thoughts of building mRNA vaccine hubs strategically in certain countries in Africa. What lessons can we learn for vaccines against FMD in these regards? However, my thinking would have been that for FMD vaccines, we need to start thinking of reverse vaccinology approaches as against conventional vaccinology approaches. For Nigeria, I was part of the team that developed the COVID-19 vaccine. It was a recombinant DNA vaccine that is being tested currently. What do we know? We know DNA vaccine ordinarily does not elicit sufficiently enough immune response. Are these vaccines going to be adjuvanted? With what? What then should we focus on more? The route of vaccine administration is key. Are we going to be focusing on methods of vaccine administration, for example, needled vaccines versus needleless vaccines? These are critical questions. Also, key and most important to my mind for FMD in endemic settings is a situation where we target multi-species, multivalent vaccines, irrespective of the technology we intend to choose. But it has to be multi-species, multivalent, encompassing a combo approach like we are proposing. But then, are we also thinking of the possibility of syndromic vaccines for FMD? Again, someone mentioned about the possibility of live attenuated vaccines. We must understand this, that the system of livestock rearing in developed worlds that fortunately for them are FMD free, and the same system of livestock rearing in endemic situations that are mostly in developing world. Livestock could move hundreds and thousands of kilometers in search of pasture and water. Hence, environmental stressors are always going to dampen the immune response in such animals. So whichever method we intend to choose for livestock vaccine development against FMD, we must consider the impact of these vaccines on climate and on environmental stressors, especially in endemic setting. Thank you.
Yeah, good afternoon to everybody. Thank you very much, Danny and Gisela. I think each side of the coin, you have well presented what is the limitations of the current vaccines. The way I look at the new technologies for foot and mouth is because the existing vaccine, though it is very safe and effective, the amount of time it takes for optimization when you are scaling up, I mean from 1,000 liter to 2,000 or to 5 or 10,000, is efforts are enormous and it takes a lot of time actually. Any new technology, if it enables the mass production, I mean it can able to, because several countries in the world still are susceptible or still vulnerable for FMDV, but they do not have the typical the BSL-3 manufacturing facilities, many countries. Still they do not have the access to the affordable vaccine. maybe any of these new technologies, if it's really affordable, and more as a, if not as a, the entrepreneur, if it is more as a researcher, if I look at these technologies, any of these technologies which can scale up, which can able to be covering the strain variations and all, it can able to make these the upcoming challenges, if you're able to accommodate with these new vaccine technologies, probably that is something really will be affordable. I think it's well quoted. COVID is the example. When we were at the vulnerable situation, we accepted even all technologies in the world, though including mRNA was never tested before, but it was, though there was a risks and safety concerns, but still was accepted. So maybe probably here we have much more advantage, a lot of research has gone. Maybe that new technology should be such a way that one, we can able to have the mass production in affordable way. So that is what something really may be interesting for the manufacturer. Thank you.
Yes. Of course, that it's very important to see new technologies that might solve, might be help to do more efficiently foot and mouth disease vaccine, more efficacious, and more duration of immunity, cross protection, etc. But nowadays, from the industry, what we are facing is a problem to differentiate the quality of the product on the market. For example, in South America or in Europe, the users were confident that the vaccine they are using is a good quality vaccine because the veterinary service, the quality control they have was a warranty about the quality of the vaccine. But when you go some other areas, in endemic settings, there is very difficult to differentiate a good quality product from a bad quality product. Even because the way the vaccination programs are performed, sometimes or very often is not optimal, so they cannot differentiate even when they use the vaccine, if the vaccine is efficacious or not on the field. But we don't have tools to check the quality of the vaccine in these areas. I was surprised when I saw the report from Medina, Oslo, Southeast Asia. No report of FMD, or a lot more from cattle than from swine. We are in Southeast Asia. FMD is more common in swine than in cattle. So there is a black hole there. They are trying to solve the problem by themselves, the big industries. They check the quality of the vaccine by themselves. Even some countries produce autogenous vaccines for FMD. So this attempt the industry to invest in the innovation, because when you go with a new product with better quality, you end up in this kind of market, which is, you are competing in a very bad way. So I don't know how is the way we can solve it, but taking more simple tools to differentiate the quality of the product may help.
Yeah,
Can I come in? Oh, sorry, Dan.
I think that's an extremely good point. I couldn't agree more. When I put in my presentation like below 50 million doses, if you want to make a good vaccine, it's you can actually not be competitive. Then it is also interesting to realize that the majority of all of the smaller manufacturers are far below that 50 million doses. So you could question, how do they do it? How can they manufacture a quality product? And then I dare to say, let's say, and they can shoot at me, but I dare to say, well, they don't. They don't. We have seen presentations, well, I refer again to it, from Uganda, with post-vaccination monitoring, and the result is a flat line. We have seen, we have heard this morning from Nigeria that the vaccines were actually, that they couldn't get any quality vaccines. It is an absolute absolute common problem that there's a lot of substandard quality out in the market. And then the question is, how do you demonstrate that? If you're a large international company making good vaccines, your price is going to be higher, but for many buyers, the only thing they see between a good and a bad vaccine, how do you see that? You can see the cap color, the label, and then the next question is, how do I know the difference between a good and a bad one? And for that, you have to set up a system, a test system, and in most countries, it's just inexisting. And it has many consequences because it also has the consequence that people will lose faith in vaccination because they vaccinate and they still get breakthroughs. Or I had a former colleague in marketing, and he used to say that the most expensive vaccine is the one that doesn't work. And that is true because you pay less, but you don't get the benefit. Well, I hear today a number of 13 times. So if you invest $1, you'll get $13 back because of the protection. If you can buy the vaccine for $0.50 less, but you don't get the $13 because it doesn't work, then it's a bad investment, then it's a very costly product. So the question is, how do you solve that?
David, did you want to come in?
Yeah, just to add in on that, I agree completely with what Rodolfo said, and I think the issue is, how do you do that? And we've heard about the difficulty of regulators in some of the less developed regions which don't have experience. I mean, it took about 5 to 7 years to get the concept of multi-strain dossier accepted in Europe, which has, a fully functional regulatory system. And many of the regulators in the less developed regions may not even be full-time vaccine assessors. So evaluating foot and mouth disease vaccines, which are some of the most difficult dossiers to evaluate, that's why you then end up with decisions that don't seem logical. So taking that forward, what can you do? And I think it's really finding a way of raising the standards around the world to what is currently recognized as best practice for regulation of foot and mouth disease. And I think we've kind of started to do that in EU FMD with our PQ scheme. And what we're looking for, and I'd love to hear ideas from everyone, is how can we take that, which is, agreed at the moment, a very centralized system, but disseminate that approach out to the regions? Because really that's what we're trying to do, to say, to get quality foot and mouth disease vaccines, you need to have testing, but you also need to know how that product is manufactured and the facilities that it's manufactured in. And I think if you can't do that, and people say, well, we're going to go and evaluate the quality of our products by post-vaccination monitoring. Well, you can't do that. You can't evaluate the quality of the product for post-vaccination monitoring unless you know that that product was reliable when it was placed on the market. Because actually then you're trying to mix up two confounded issues which can't be distinguished by serology. And that is, was the vaccine any good in the first place? And secondly, was it applied in the right way? Was it appropriate? And so on. And so, you look at South America, when they do PVM, they don't have to worry about the first issue because they know, and they've checked the quality of the vaccines. They know that if there's a failure, it's not because when that product came out of the manufacturer, it wasn't any good. Something's gone wrong beyond that. And in most other regions, you can't do that. So it really is focusing on what are you trying to measure, the quality or the performance in the field. Thanks.
I have a question here, sorry.
Okay, and then Keith after
And
this
then
one.
Keith, and then here at the front.
So I don't have a question, I have a comment from a manufacturer's point of view. Coming back to Toby's original questions earlier, is as a manufacturer, there's nothing that we need extra to make new products and to invest in new technology. I think from the amount of posters that we've seen during the session, it's clearly indicative that companies are looking into making new technology, making new products. That urge is there. The problem rather is time. And coming back to our colleague from Nigeria, what can we learn from a COVID vaccine and the COVID pandemic? We need a human pandemic to get people to reduce the level of evidence that's being required after making a new product, because I'm not saying we have to do it, Aldo. I'm saying that is what the reality was. We had to get a product on the market exceptionally quick to stop human deaths. That is not the case for FMD. FMD is an endemic disease. Nothing is changing. So the level of evidence that is required is going to stay the same. And that requires time. And there's nothing that manufacturers can do to quicken how long it takes to do an animal study. So it would be ideal if we can get new technology onto the market in 2 to 3 years. It's just not possible because we have to do the normal processes that all of the different regulators have set out there for us to do, because we all want good quality vaccines. We want to know that the product is working. We want to know that the product is safe, and therefore we need time.
Thank you very much. Now, Keith.
Thank you. I go back to Toby. I think you posed the question about endemic regions and what vaccines might be the most appropriate eventually for those regions. I would have thought that duration of immunity would be probably one of the most key selling points and valuable in a vaccine for endemic settings. The idea that you might vaccinate even in the presence of maternal-derived immunity from a very, very early age and it would last for multiple years, I think would be a game changer. So we could ask, which of those ones we've heard about? My sense would it be, from what I know, limited immunology. It might well be modified live vaccine that would provide that. So while it might be regulatorily very, very difficult, perhaps longer term, those duration of immunity and ability. So question almost to the research community and those people who really studied the literature for their PhDs and so on is, what is our evidence of duration of immunity after natural live infection? Many of us assume it's about 3 years or something like that, but actually I don't think there's very much evidence. So some of you in endemic settings, I mean, think about, as you go home, think of a nice experiment where you buy some animals that have been recently infected, or make a deal with somebody who keeps them for a few years, as far as you can tell, free of FMD, and then see if someone can challenge them. Anyway, this is researchable, but what is the natural immunity? And my sense, and I think other people might say, well, you're probably about right, will be about 3 years. But it might be 5 years, it might be longer, it might be most of the life. Viruses change, but maybe the immunity to that strain doesn't. So duration of immunity, I think, would be one of the most sellable points. And maybe those who are involved with the new technologies can propose, apart from modified live, which other ones would have potentially the longest duration of immunity.
Thank you, Keith.
Well, if you could also ask yourself, what is the duration of immunity of a live polio vaccine, knowing that polio is very closely related to FMD. I think most people will agree that it is a lot more than 3 years. I mean, that's maybe even lifelong. So why would, for live FMD, the duration of immunity not be considerably longer with the live approach? I think it's a very relevant question because it would make an enormous difference in the field, of course.
I think Pascal's waiting for next.
Unless you want to finish on Keith's question, which is really to the point.
Okay.
Yeah. Continue with the duration of immunity.
The question is about my colleague from Nigeria touched the point when he talked about the potential factors that may interfere with the vaccines in endemic areas. And here I'm coming back to the situation of countries where we may have comorbidities. Here I'm targeting mainly tick-borne diseases. And here I'm thinking about Congo and other similar countries where we have a high prevalence of tick-borne disease like ECF, East Coast fever, babesiosis, pyroplasmosis, and anaplasmosis. So I'm thinking that during the development process, it would be better to also consider exposing animals in that kind of areas as well. So proceeding by multi-sites research, where some sites may be installed in such places of the world where you can see how animals can develop immunities if at the same time they are facing tick-borne disease like like ECF and so on. I am drawing from my experience when we tried to introduce ECF vaccine in Eastern Congo using Muguga cocktail vaccine for those who know it. We tried to use infection and treatment method. That was the method for the vaccine. But we saw almost the same rate, the same mortality rate in the vaccine and non-vaccine groups. So there was a lot of confusing factors during the exposure step of the process. So thank you.
Yeah, what you're saying, in fact, we have done some experiments infecting animals with fasciola hepatica, and we find that the immune response in those animals using the conventional vaccines is diminished, it's changed, and has not enough antibody levels related to protection. The same happens with the animals infected with leukosis. So yeah, I think that what you're saying is true, that all the vaccines are always tested in healthy animals, and some areas have underlying conditions that can affect the immune response. But I think he has a comment.
There was a comment to what the community can do, what all of you can do, maybe not personally, but through your CVOs, I don't know. It's two things that we heard already, but are really important. The multi-strain registration framework, put it in the WOA manual. When you upgrade, when you revise the FMD monograph, put it in there. It is the reference for many countries that do not have a national set of standards. So if that could be put in the WOA manual, that would be a great improvement and much easier to develop new vaccines. And the second one is Nagoya. This is going to be a big problem for all of us, and not just for inactivated vaccines. Huh? It will apply to sequences. Some countries already have sequences in the scope, so it's going to block the development of new technologies as well if we don't find a solution.
Yep. Owen?
To further comment on the hurdles, I agree with Pascal that there are regulatory hurdles, especially outside Europe. But I think one of the biggest hurdles that we, for instance, face with our technology is FMD is just very complex. And it's not just one vaccine for one species. It's multiple serotypes, multiple strains from multiple species. So if you want to demonstrate the efficacy of a certain strain, you have to do the efficacy studies in goats, in sheep, in pigs, in cattle, as well as safety. So if you want to build up a portfolio, and you have to, because you know you want to serve every region with a proper vaccine, you end up easily with 40 trials or many more. And that's all okay. I think we all want to know, even we as a producer, everyone wants to know that the vaccines are safe, so we have to go through it. I agree with my colleague. There's nothing we can do about it, and that's also the reason, for instance, why COVID could go fast. One antigen for one species. It's a bit easier. And I wanted to say something else, and I've forgotten about it, but so I give the microphone back, Aldo.
Yeah, Vilja? While you're preparing your question, I mean, so one question asked at the start was, just to sort of reiterate the question, do we need to develop novel technologies at all? Or if we just made conventional vaccines globally that were good, would that do the job? Yes. Well, we know your answer already.
You said yes.
Vilja?
So here's a thought. What if all the vaccine manufacturers get together and work together and make sure that these things can be done fast and quick and then go out and serve the world, instead of every vaccine manufacturing company working separate on the same things, facing the same issues, the same costs, and maybe things go just slower?
I think
same strains.
I think, Vilna, I think it's
They don't
easier
like
if
each
all
other,
the regulatory authorities got together and agreed on a global situation, so you only had to do one registration dossier for the entire globe. I think that would be more likely to be successful.
And just to say on that, I mean, unfortunately, the history of regulatory cooperation in the area of vaccines is not brilliant. If you look at VICH, there are around, just off the top of my head, about 20 VICH, that's the regulatory cooperation group, guidelines for medicines, and about 4 maybe about 6 for vaccines. And it's just because the philosophical approach of the 3 main areas, Japan, Europe, and US, has been very different. And so it's taken many, many years to come together. So if you're looking for a test for mycoplasma or residual moisture, you can get a VICH guideline. If you're looking for efficacy, well, there are some safety ones, but it's traditionally been very difficult. But I think that's where something like foot and mouth, you have to start small. There's a very clear need. It has been, the kind of regulatory problems have been cracked within several regions. And then it's spreading that best practice and building the confidence of regulators. Because I think the point Erwin makes is very, very good. Foot and mouth is one of those vaccines where you absolutely have to extrapolate data, say from one species to another, from one strain to another. And that's something that regulators are very, very uncomfortable with. You know, they say, well, you've shown it for this strain, now you have to show it for that strain. But look at foot and mouth, you know, if you've got to show it for every strain, for every species, for every category within that species, it's impossible. And so it's really getting them comfortable with the fact that actually you can extrapolate that data with minimal risk, and we managed to do it with the multi-strain. And I think that suggestion of getting that multi-strain and opening the kind of world up to that concept, now that it's been developed, shown not to be a regulatory risk, would be very valuable. Yeah, thank you.
So these are my observations on the FMD vaccine, which is a victim of its own success. Ever since 1965, Capstick and his co-workers built the BHK substrate to grow FMD. We've not found another substrate to grow FMD. And as you showed in your presentation, you need 10,000 liters of cells to produce certain number of vaccines. That means you are using a very poor substrate compared to a PPRV vaccine, which can be grown in a monolayer, if that is the comparison to comparison. That means we were successful in making a substrate to grow FMD. We had the procedure to inactivate FMD virus. And then what about the adjuvant to increase the duration of immune response? We are not talking about that component of the vaccine. We are talking about the vaccine strains, vaccine subtypes, serotypes, whatever changes we are having, the payload and those things. But 50% of the component of the vaccine is the adjuvant. And we are not considering anything of the adjuvant because we are happy with the so-called oil adjuvant and now the new alum adjuvant. So when are we going to think about the adjuvant to prolong the duration of immunity? Thank you.
So I'm aware we jumped straight into the live attenuated kind of debate right at the start, but I mean there's a lot of other emerging technology. So if people have comments or discussion points about, yeah, Owen.
I remembered what I wanted to say. Thank you, Toby. It's about live strains. I think there could be a potential there for emergency vaccination, maybe in case of outbreaks. But for endemic regions, I doubt it's the way forward, and for the following reasons. Co-infections, I think, do exist in foot and mouth, but if you would want to protect against an O, an A, an Asia 1, a Z1, Z2 serotype, you need a cocktail of 5 live strains. What will happen in the animal? Can you really get an immune response against all 5 strains? I doubt it, actually. So this is something that
worked for polio, didn't it?
Yeah.
Okay, so maybe that's, I'm not up to speed to that, but this is something we need to find out whether this is possible. Yeah, that's I think my greatest worry. And the other worry I have with live attenuated strains is, what happens in animals that have been pre-exposed, either by vaccination or infection? Will the antibodies in those animals clean the vaccine strains before they can do anything? That's less of a problem with an inactivated or subunit vaccine. So that's something we need to study, I think, before we really get very enthusiastic about live attenuated strains. But that's just my personal take of it.
Okay. Any other comment?
Yeah.
So we should focus on new vaccines, or we stay with the same? Or you comment on his?
Yeah, just to answer Erwin's question. Of course, if you use a live attenuated or a live vaccine, and in the presence of antibodies, whether it is maternally derived or whether it is from previous vaccinations, that you won't have an extra effect of the live, or very minimal anyway, because your live vaccine is also going to be neutralized by the antibodies. But that's for every live attenuated vaccine which is used. That's not specifically a problem for FMD, I would say. On the other hand, if it will neutralize the FMD vaccine, then it will normally also, at that moment at least, protect against infection, I would say.
And the inactivated vaccines also have problems in maternally derived antibodies.
Yeah, absolutely.
So it's not a difference.
So just to conclude, I guess, the discussion about the live attenuated vaccines, and I mean, the landscape that we're in with FMD, I think we should all be aware, of course, that there is some history with FMD, and one of the strains within serotype O that circulates in the Far East, in Paul 1, probably has come as a result of an attenuated live experiment that didn't work well. So this is the famous Cathay strain, the O Cathay strain, is probably derived from an attenuated live vaccine that was made sometime in the past. So that's, I think, that's the space we're in, and I think we have to, that will be the criticism, I think, when these products are taken forward, is to prove that that won't happen again.
Can I have a question?
Yeah.
I have a question just for the companies. We are talking about one of the advantages of the live attenuated vaccines to extend the duration of immunity, but I'm wondering if companies are exploring, for instance, adjuvants or additives to the formulation that could extend the duration of immunity beyond the 3 months or 6 months that we have in the standard. Is there opportunities for research or trials on that? And if so, is anybody looking into that from the company's perspective?
Oh, hi. We are from CDB, Argentina. We are from the Research and Development Department, and yes, we are working on different adjuvants in order to improve. We don't have all the data yet, but we are working on that because we think that is also very important, not only the quality of the antigen. So we are doing a lot of experiments with that. But one of the problems is that everything takes a lot of time in order to do those studies. You need a lot of time because you vaccinate and you have to wait for a long time in order to get the results. So it's complicated to move quicker in taking that into consideration. And also, what we were wondering with the live attenuated vaccines is what about cross-protection? Because if you can develop a system in which you have cross-protection within different strains, it would be easier to register a dossier in maybe one country and be able to move on to others. If not, you have to start changing all over again each time for each country, and that is very complicated, and also it's very expensive, and not all. For example, we are from a third world country, so everything gets a little bit more complicated regarding the regulatory affairs and all the experiments that are needed.
Thank you. I think we've got a comment by Aldo. And is that?
No, just
and then after that, I think we should start wrapping up for a coffee break.
question from Lisa. In South America, we worked for many, many years to produce vaccines with a long duration of immunity. In fact, we need to demonstrate in the licensing in any country in South America that with one shot you can protect at least 6 months, and after the booster you need to protect 1 year. So it was a big challenge. Most of the industries they went through, they solved this problem. The supplier of adjuvants during the time they were evolving, there are new alternatives. You need to check, you need to see if you need to develop a new product, or you can adjust the product you are producing. Sometimes the adjuvant changed because some people here might remember when they changed the olein because BSE outbreaks. We got to change all the things in the adjuvant. But South America has a lot of experience producing vaccines with long duration of immunity using single oil system formulation vaccine.
Thank you.
Perhaps I should add that the Netherlands produced a Frankel vaccine up to 1991, and we only vaccinated once per year. A young stock only after 4 months, only 1 shot, and then the next year round, and we still were protected against the introduction of FMD, and had very good antibody responses. So the short duration of immunity, which a lot of scientists say, is not based on good literature review, because a lot of papers showing that there is more than 6 months high antibody titers.
Yeah, it also depends on what you measure, because the presence of antibodies, it doesn't mean duration of immunity. There cannot be antibodies anymore, but you have memory cells, and you challenge the animal, and the animal is protected. And we have done that experiment in South Africa with the group of Francois at the time. And we waited until the antibodies waned, so they were really low, and then we challenged, and animals were protected. So we need also to define what do we expect for long duration of immunity. Antibodies cannot stay forever if you don't get the antigen again. So you need to boost just to see the antibodies around. But that doesn't mean that the animal is not protected, because you can have memory cells. And in fact, the infection is a booster, as it is. So that's something we need to define. Yeah, we have someone there, and then...
Yeah, this is still a comment to the live attenuated vaccine. I mean, we heard several times today how important the quality control of the vaccine is. And I guess one concern, if we allow the framework for live attenuated vaccine, is that reassurance for the quality control becomes even more critical, because what is the effect of a poor quality live attenuated vaccine? I mean, lumpy skin disease just showed us the issue, what could happen if a vaccine gets on the market which doesn't quite comply with that quality control. And I think there's just a bigger pressure that the quality control for live attenuated vaccine would need to be fulfilled.
But if I may answer, a quick answer, but that problem, I would say, doesn't lie in the technology, that lies in the control or in the question on how do we control all these local manufacturers who cause these type of problems, because there's no proper master seed testing. But that is a different problem, and you're going to get that anyway if you allow them to make live attenuated vaccines. Whether we talk about FMD or something else, it's no different.
Mariano.
No, just to add what Alejandra said. That's the reason why you can find protection with very low level of antibodies, not only at long times, but at early times as well. So Of course, the immunology ways are mysterious somehow, and they are not always measured by the conventional methods, but somehow vaccines work, and it's not always that obvious how they do that.
Thanks, Mariano. Okay, so I think we're gonna pause there because we have a coffee break scheduled. Thanks to everyone for the contribution to the discussion. That's been really good. So we're gonna have about a 25-minute coffee break. If you've got additional discussion points, keep them in your mind. We will, when we come back from the coffee at past. Yeah, Aldo, I think that's mine.
Oh, why didn't you say that?
So we'll come back at past. I've been waiting to say that for quite a while. We'll come back at past, and we'll do a wrap up of the discussion forum, and there'll be time for a little bit more discussion if people have some more burning issues that they want to raise. Okay, so thank you.